and thank all of you for your different roles in making this a very exciting and engaging meeting. From those of you who built the beautiful auditorium and the beautiful institute, to those of you who are performing the extremely important role of sitting there in the seats with your ears open. Nothing harder than giving a talk without an audience. So I have slightly recast the title from the published version up here to emphasize the open question I want to emphasize here. So let me walk you through it. The subject which I will hold forth on at length is diversity of synapses, functional, molecular, structural, in the brain, let's say. Let's say in the cerebral cortex, an interesting place to think about. Okay, so that provides the background. Now, the question, the open question is friend or foe? And I don't want to keep you in suspense too long. I will tell you what I think about this question. I think right now, synaptic diversity is a very formidable foe that undermines everything we're trying to do in understanding the brain at every level. And the reason for that is that we don't understand it. And the reason we don't understand it is we don't have the right tools to shed light on that particular subject. On the other hand, I think those kind of tools are just around the corner, and my lab has been working pretty hard on one candidate tool. We haven't broken the back of this formidable foe yet, but I will spend most of my time telling you what I think we can do about it with this one technique. Other techniques will certainly be necessary. I think once we learn enough about synaptic diversity, it can be just as powerful a friend and aid to everything we're trying to do as it is currently a foe. So there's my speech about open questions. Now to dive in. Actually, we'll dive in. I'm just going to put this slide up for a second to set the tone. Up here we have one of the many metaphors for this activity that most of us are engaged in of trying to understand brains. It's a metaphor not without its weaknesses, not without its strengths. It's a fairly current one at the moment, and I think it's a useful way of framing my thoughts about synaptic diversity, why I in particular care. I like the idea of understanding how things work in terms of both their structure and function. In fact, I think that's what understanding how something works means. Now, the folklore, and I'm sure this happens occasionally, I don't know how often, is that in the world of industrial espionage, people get their hands on one of these things, and first they do a bunch of anatomical experiments on one of them, and then they do a bunch of physiological experiments. They use fib-sims for the anatomy, logic analyzers for the physiology, and this is what they're up against. These things are pretty complicated. Also, actually, it is rumored that the people who make these things put spoofs and tricks to waylay people who are trying to reverse engineer them. We know evolution would never do a thing like that to us. On the other hand, there is the slight advantage that as far as we know, these things are always designed by humans and that the rules are well known and published in all the textbooks. Now this is the thing that most of us in this room are more interested in directly cracking open. And part of the problem is similar. And we will hear about this. You will hear about it in other talks. This is maybe the connectome, getting the wiring diagram, the connectivity matrix. It's important in both contexts. But what I've tried to do here is emphasize a dimension of the problem that is absent over here. And when you think about it, I think it might be the big problem in reverse engineering one of these things. And that has to do with the complexity and diversity of the components. Basically, there's hardly a gene in the species in question that you can ignore completely when it comes to thinking about brains. Most of them are expressed in there. The Allen Institute, of course, has done a beautiful job of looking at the patterns of the expression. And one of the things that comes out of that is there's lots of genes expressed in the brain, and their different genes are expressed in different cells. So, that's the roots, let's say, of the diversity problem. So, there's a big diversity problem which is absent over here. There's like 4 things you need to worry about in a typical chip, and, by the time you put in splice variants and stuff, who knows over here. Okay, so we're going to zoom in. I'll try to get to my point about synapse diversity. So here's a lecture, a 1-hour lecture in itself, but on the other hand, material is familiar to both of you, to most of you. This is what I mean by synaptic diversity. These are some of the dimensions of it. There's molecular diversity, different matter in the synapse, structural and functional. Today, we will concentrate on this part. They're all important, and building the bridges between them someday is the reason for doing any one of them, I think, especially in that reverse engineering context. So this is a list of all the different kinds of molecules that are in a synapse, and Seth Grant's talk yesterday was a good introduction. He took one little part of the synapse, I think this part represented over here, the postsynaptic density, and talked about how incredibly complex it is in terms of the number of molecular components that could be there. And then in the questions, it arose that the degree of diversity, are all of those molecules present in every synapse, was not clearly understood. That's the subject I'm talking about, the synaptic diversity. And I'm not going to read these off, but it is a complex machine. These are little snippets. Here's the PSD over here. But actually, you can break it down. A particularly interesting part is the synaptic cleft itself. There's a huge number of diverse molecules there, some of which we think about as signaling between presynaptic cell and postsynaptic cell. Most of them we think about as being the cues that wire up the networks of the brain in the first place. These are the adhesion and guidance molecules that help build those incredibly intricate circuits that compose the brain. And as we learn more and more about them, there's more and more molecules there, and a beautiful paper out of the Allen Institute a couple years ago showed that the patterning of the expression of, like, for instance, the adhesion molecules in the synaptic cleft is for sure extremely diverse on a neuron-to-neuron basis. We don't know, how that breaks down to a synapse to synapse. There's canonically something like 10 to the 4th synapses per neuron. Are they all the same? Well, couldn't be probably, because they're between two different cells. But anyway, that's one of the kinds of questions. Okay, so to me, it's a giant topic. It's one that we haven't worried about too much because it's really not a good idea to worry about something you can't do anything about, by and large. So why is that? And I think one of the main reasons is that the tools for looking at synapses or synaptic transmission on a single synapse basis have been extremely few and far between. There's a standby that is, applied in industrial quantities, and that's, counting up dendritic spines. It goes back 120 years. It's part of the birth of neuroscience. It's where the concept of the synapse came from. But, from our current perspective, where we know molecules matter and coefficients matter, it doesn't really tell us that much. On the other hand, the well-known industry of counting dendritic spines tells us what a precious thing having any surrogate for the individual synapse is. However, very limited from our perspective, basically no molecular information. Electron microscopy has, for the last 50 years, has very richly enhanced our view of the synapse and what it is. However, with the electron microscope, even with these new high throughput techniques, people have looked at, a few 100,000 synapses. And most of the electron microscopic study, indeed, it isolates single synapses, but it only gives you a part of it, and it's a huge amount of work, and it's slow, and there's very little molecular information. So those are the two established single synapse methods. Very much nearly end of story, except perhaps in isolated cell culture situations. Physiology, well, I don't have time to try right in this, but most of the things that you think of as a single synaptic connection are actually parallel arrays of tens, hundreds, thousands, in some cases 10,000 similar synapses. The calyx of Held is a parallel array of synapses. Maybe they're all the same, maybe they aren't. Okay, 3 question marks. Proteomic imaging. Well, we don't have much proteomic imaging, but I'm going to tell you about a way, and I think this could begin to build the framework to start really attacking these single synapse type questions. So very briefly, this technique we call array tomography. It is something that gives you three-dimensional volumetric visualization of a tissue at high resolution, at a resolution that's about 50 times higher, for instance, than the best confocal. That is in a volumetric sense. The voxel is 2% the size. So it's very high resolution. And that the significance of that 50-fold improvement in resolution is that array tomography, at least in mouse cerebral cortex, readily resolves individual synapses, individual synaptic puncta, pre from post. It doesn't resolve their very finest details. But for instance, what you might think would be the closest competitor, the confocal microscope, has a whole lot of trouble resolving individual synapses, especially one that's above or below. Confocals have terrible axial resolution. Array tomography is proteomic. Right now, we're looking at dozens of molecules per protein punctum. It's not clear that that's not a theoretical upper limit. It could be hundreds next year. This is important. It is, even though it's based on immunostaining with antibodies, and it's the form we've applied mostly, it is inherently depth independent, unlike most antibody immunofluorescence images, and therefore it gives you an enormous quantitative hook on your system. You might not know the number of molecules your antibody is detecting, but you can compare them quantitatively from one piece of the brain to any other piece of the brain in 3D. I think a very important thing. It also has an elegant capability I don't have time to talk about for doing correlative or conjugate light microscopy and electron microscopy. Electron microscopy is the ground truth for knowing a synapse when you see it and for a lot of important questions. It's sort of a mashup of lots of different old techniques, mainly techniques from electron microscopy, glued together with a ton of modern computation. The brief description. There's an automated serial sectioning process. You can see neat little ribbon of serial section being produced here. Those get placed onto cover slips and built into a thing that's kind of like an Affy gene chip for those of you who know those things. It's got a nice little window you can image perfectly through with a high NA objective and keeps it nice and clean. You take your specimen and you do what we typically do is standard 4-color immunostaining, and then we put it on the stage of an automated microscope, and here it is. The microscope in real time takes a whole bunch of pictures, and you can make big mosaics and do very large areas. Then, well, there's computation rearing its head right there. Then we use computational alignment techniques to stitch everything neatly into 3 dimensions. So that's the quick, and we've got papers out. There's actually a very nice new paper coming out next month. We've published lots of protocols. I love to talk about it in detail, but I think I better move on given, oh dear, I forgot to start the timer button. Maybe somebody can figure out.
I'll let you know.
Yeah, okay, yeah, let me know. Okay, so we've got a problem and we've got a tool. We better start putting it to work. And in a not completely arbitrary decision, and perhaps Karl Svoboda will tell you more about what's so cool about this particular system later today, we decided to study the cerebral cortex of the mouse, in particular the part devoted to whisker sensory motor connections. It's a very popular part of the brain for physiologists to work on. The best are flocking to it, and it has a lot of inherent advantages. For the moment, suffice it to say, it looks like this. This is the icon we'll use for navigating it over and over in this talk. So this shows a sort of coronal section through barrel cortex. Here's the scale, 1.5 millimeters. The cortical mantle is about 1 millimeter thick in the mouse. It's layered. These have been described for a long time. And partly because they just look so cool. We will be making extensive use, we are making, of transgenic mice that express YFP. And this is probably the champion all-time workhorse, the Thai1 line H YFP mouse that you've seen in so many in vivo 2-photon studies came out of Jeff Lichtman and Josh Sain's lab in the waning months of the last century, and it was primarily the work of Woping Fang, a wonderfully useful little work mouse. Okay, so this is a thumbnail sketch. There's the layers, surely familiar to many of you. Okay, now if we could dim the lights. Okay, here's our little navigating icon, and I hope you can see this little magenta rectangle, and we'll use this over and over again. The magenta rectangle tells us where we're zooming in and looking at an array tomographic volume. So, you know, part of the point is it's a high-resolution volume image. And so what you're seeing here are the subset of axons and dendrites that happen to be expressing YFP in the line H mouse. And by and large, and we're up in sort of layer 1 and 2, by and large, the big things and the things with spines on them are the dendrites, and the little things, the little spiderweb threads studded very often with little varicosities along their length are the axons. And so these big celery stalk dendrites you see are from big pyramidal cells down in layer 5B, and they're projecting up here into this. These are the large tufted dendrites, and there's their, you know, mostly their dendritic tufts interweaving in the beautiful and enigmatic layer 1. And then there's axons, and some of those axons could be local recurrent collaterals of the same neurons that we'll see later, and others could be coming from the other hemisphere. They're long-distance projections, terminal arborizations, and at this point we don't have good ways of being sure which are which. Okay, so now another thing that I will just point out, this is a good place to point it out, you will periodically see axons and dendrites that appear to just abruptly start or stop. Like, it's very unlikely that that dendrite terminated bluntly. It kept going and made a beautiful arbor. It's just that this volume is only about 15 microns thick in depth. And you know, we tried our best to cut it in the plane where all the dendrites were growing, but of course they don't all grow in the same plane. So you know, very often you'll see something that enters in the back of the block and leaves the front of the block. On the other hand, when things are within the block, which you can tell with 3D navigation, they're beautifully, continuously mapped out, even these little axons, which in truth are only 50 to 100 nanometers in diameter in a lot of cases. Oh, yes, thank you. Actually, in a way, the answer is on the next slide. So let's just look at this while I answer your question. So this is 3 or 4%, in the line H mouse, 3 or 4% of the cortical neurons express YFP. It's a particular subset. It's layer 5B pyramidal cells. The other neurons, the other dendrites, the other axons, the other 97%, let's say, do not express YFP. So in most of the renderings, you'll see invisible empty space. That invisible empty space is actually packed full of other dendrites and axons, roughly like the ones that you will be seeing. It's just that there's 30 times as many invisible ones as there are visible ones. So this is throwing in an immunostain for acetylated tubulin. Axons and dendrites have lots of acetylated tubulin in them. It's important to allow them to do what they do and stay alive. So this gives you a sense. You know, the tubulin doesn't give you the kind of beautiful resolution that the YFP does. It doesn't light up spines so well, but it gives you a sense of how much stuff is really there. So thank you for asking that question. All the stuff that appeared as empty space is actually full. Now, even so, in this tube, actually, it'll be better in. Okay, now here's another look. Now we've stained a synaptic marker. It's synapsin 1. Our paper next month will argue that synapsin 1 is actually the, if you want to look at just one molecular marker in cortex, synapsin is the one that comes closest to showing you all the synapses and only the synapses. There are plenty of markers that are present in all the synapses, but they're also present in lots of other places, and it just so happens that synapsin gives you the best rendering. So now you can start to see little individual puncta. We still haven't zoomed into the full resolution, but you can already begin to get the idea that they're individually resolved. And probably some of you are looking at the spines to see if there's a little red cherry on the end of every one. There is. I'll show you that better next. But anyway, another generic feature of all the volumes we'll be looking at, that I just want to point out, is there are these little pockmarks, these little cavities sprinkled around. Those, by and large, are cell bodies. Actually, some of them are blood vessels, too, capillaries. So there's capillaries and blood vessels. You'll see some obvious capillaries later. But anyway, most of those little voids you'll see, and you'll see them in all of these renderings, are cell bodies, because guess what? Cell bodies do not have synapses inside them. They do have some synaptic markers, but not much in the way of synapsin 1. OK, so now we just continuing our tour, we'll go down to layer 5A. And down here, we get, now we've kind of there's a layer 5B is more than 3 or 4% here. So this enriches things a little bit, but it still drastically underrepresents the population of what's there. This is a little higher magnification. And now, you can begin to see the individual dendritic spines a little better. And this is just a sense. I mean, we've known for quite a while that there's between 1 and 2 microns, synapses per micron. There's a lot of microns, even in a mouse brain. This turns into vast numbers of synapses. In a way, that's why diversity is important. Synapses, are believed to be the most important signaling element in the brain, the place where memories, one way or another, are expressed, either the presence or the absence, or the motivational state of an individual synapse. OK, so finally, here's the home of the layer 5B pyramidal cells, up close and personal. And now you can see a little cell biology at work. You can see intermediate processing or degradation forms of the YFP and weird little things in the cell bodies. And you can now begin to get a more clear, we're at a little higher magnification, the resolution of individual synapses. To drive the point home a little farther, we use just simple 3D image analysis and segment out all the individual synapses. And here, each synapse is digitally segmented out and assigned a random color. So this is a pseudo-quasi-brain synapto-bow mouse in the technical jargon of the field. But this drives home the point that, yes, we can resolve, segment, treat, look at, analyze, wash, and dry individual synapses. OK. So here's, zooming in real close, here's one dendritic spine, some synaptic markers on it. And I won't belabor it, but they're the right synaptic markers. Every spine has a postsynaptic marker. It has some glutamate receptors. It has some PSD95, a cast that Seth introduced. The state of our art right now is we can routinely do 18 to 24 markers per synapse, which we do by repeated cycles. We do 4 colors at a time, and then the plastic matrix in these thin sections stabilizes the tissue. We use a pH change to strip off the first 4 immunostains, primaries, secondaries, both, and then we put on 4 more. And you can do it over and over again with the same markers. You get the same answer. On the other hand, you can use that to multiplex lots of markers. Now, the trouble is, the point I'm making here is you can't actually, it's kind of hard to look at 18 colors at once when they're all mingled together. It's fun, it's pretty. The simple analysis that we use all the time in the lab is something we call a synaptogram. So this is an array of, this is 2 synapses, 2 little 1-micron cubes. So there's 9 serial sections centered around, in this case, the local maximum of synapsin 1 immunofluorescence. And then down here, 18 different molecular markers. And the point is that we believe with this kind of approach and the right panels of antibodies, we can determine the molecular, we can begin to crack that molecular diversity. We can begin to build a molecular catalog of synapses, we hope. We aren't there yet. We're doing various things. I mean, we're doing some unsupervised clustering, and this just shows that 3 well-known classes, vGlut1, vGlut2, glutamatergic and GABAergic synapses cluster out pretty nicely, just with their molecular markers. We're using to pull some numbers out, we're using a machine learning approach. Very powerful stuff. So here's the really quick take on it. So there's some data. Too many markers. Nobody wants to count them. Think about where they are exactly. So the supervised machine learning, we begin by just, we enumerate the synapses, then we use a random number generator to pull out some random subset that we just might be able to coerce a human expert, i.e. a graduate student, to spend a couple hours classifying by hand. So they stare at some of those synaptogram things. Here's one. The human expert looks at it and notices it has some glutamatergic markers and the GABAergic markers are absent. So the student pushes the button and calls that a glutamatergic synapse. And the features extracted from this little volume are stored away in the computer somewhere. And then here's another one with some GABAergic markers. Student pushes GABAergic button, etc. So here's the result. Here's the training set, hand classified. Now the reason for doing that is there are a number of standard machine learning algorithms where you put the training set in, it infers a set of planes in a high-dimensional space and makes it so it's no more work, at least for a human, to go through and classify the other 10,000 or the other 10 million synapses in your data set. And so here's little idealized balls where we've made the classification based on, in this case, a set of about 10 markers of inhibitory versus excitatory. And you neuroanatomy wonks will recognize there's some things that look right here, like lots of GABAergic synapses around the cell bodies. Once you've done, it's fun to look at those pretty pictures, but it means you can start to pull out numbers. And there's sort of a natural, one of the many things we like about the cortex is it has kind of a natural spectral axis, depth below the peel surface. So here's some numbers peeled out. You can also use 3D image processing tricks. If you have some subset of cells labeled with a viral tracer, with a dye injection, or with a transgenic, like in this case, good old line H, you can use 3D image processing tricks to segment out individual neurons of somewhat known type, somewhat known projection origin, and then use more 3D image analysis to pull out and look at the synaptic markers that only have just the right relation to that are real close and have their postsynaptic stuff closer than their presynaptic stuff. Good guess that any such neuron, synapse, would be a synapse onto that cell. So this is a way of cutting the synaptic diversity. We can say, what happens, how does the synaptic diversity everywhere in layer 4 compare with the synaptic diversity only of synapses onto layer 5B pyramidal cells in layer 4? Just an example, and here's a little machine vision classification and pulling the numbers out of that one. Okay, now, here's some stuff that would be fun to talk about, but we don't have time. I hope you ask me what the heck a synaptome is in the questions. What I actually want to do is I want to show you what I believe is a new high water mark in supplemental materials in journals. This will be supplemental video number 1 from a paper with that list of authors and title that will be coming out in Neuron, November 18th issue. And one of the things that's so special about it is it has an original musical score by this extremely talented young composer, singer, songwriter named Catherine Rose Smith, who happens to be my daughter. This is a father-daughter collaboration. What could be better? Okay, so now I heard the sound is working, and you really need good speakers. It says in the supplemental materials, you need good speakers turned up loud for this one. I think it's that's the first time that phrase has ever appeared in Neuron. Okay, now we could have the lights down, and maybe you can keep the sound up about to the threshold of pain. So I think you know what you're seeing now. This is a tour through the machine that created neuroscience and math and physics. Well, the mouse version of it. But the human cerebral cortex has got to look a lot like this. It is a magnificent and beautiful machine. An important thing to keep in mind as neuroscientists, we are studying the most beautiful thing in the universe. Actually, Catherine is going to be very thrilled when I tell her that her music has now debuted at the Experience Music Project. She is, this could have been the inspiration. She is now a freshman in the Ultimate School of Rock, the University of Southern California's new popular music performance program. You know, I wouldn't be surprised if she doesn't help out with my comfortable retirement, but you never know. But it could be that the time I took her here when she was 10 years old inspired her in this direction. We went up this little family thing where the family can, you know, play music up there. We did that. Okay, thank you.